So good afternoon. My name is Misti Ushio. It's a pleasure to be here. Thank you for the invitation. And today I'll tell you about Tara and how we're engineering human biology to transform cardiac drug discovery. I wanted to just touch on a couple of things today. One, a little bit on my background over the past 30 years, kind of where we are today with Tara, and give you a flavor of kind of the early formation and company building, and sort of the growth and things we considered along the way. So just in terms of my own background, I have a bachelor's degree in chemical engineering and a PhD in biochemical engineering. I worked at Merck for over 10 years in vaccine bioprocess R&D, and so we were developing the manufacturing processes for the biologics. I then moved to the tech transfer office at Columbia University to really kind of think about how do you start to translate, cutting edge technology coming out of academic institutions, into companies, into commercial opportunities, which then led me to a venture capital group here in New York City, focused on early stage venture investing. And so had that experience for about 10 years when I learned about what is now the Tara technology. Initially, Tara was an investment thesis around stem cell technology and tissue engineering. And it really was kind of the foundation to kind of where we are today. And so kind of how do those different experiences and how has some of that translated? So at Tara, just for those of you who don't know, we really focus on generating the highest fidelity in vitro biology, and generating, human biology. We can generate human relevant data. And a couple of facts along the way. We were founded in 2015. Our scientific founders were Professor Gordana Vunegnabakovic at Columbia University and Professor Melissa Radisek at the University of Toronto, who I believe many of you know. And they had been collaborating for many years and met at MIT and Bob Langer's lab. We raised about 20 million to date and have over 23 employees. And so why do we do what we do? I don't have to tell you guys that, heart disease is the #1 killer of men and women worldwide. It's a big problem. And still, drug discovery and development is really long, it's very expensive, and the probability of success is very low. And moreover, for cardiovascular drug discovery and development, the probability of success is even lower than other therapeutic areas. And so it's not for lack of trying, we're trying to move and we're trying to develop new medicines to get to patients, but it's hard. And why is it hard? Well, partly because we don't have very good predictive models. We have simple cell assays, we have animal models, which are not human. And so we really, are lacking information before we can get into clinical trials and, which is where you see a lot of the failures. So we're really trying to increase the probability of success for cardiac drug development. So we're able to engineer models of human heart and human heart disease. And we do this by leveraging the advances in stem cell science and tissue engineering. This allows us to accurately measure the efficacy of novel molecules and their effect on heart function. We have been working extensively with pharma companies over the past 4 years, which has given us a really nice view into the application space and the validation of the platform. And as I mentioned, we're really focused on how our technology can increase the probability of success. You can really get new medicines to patients. So, taking a step back, what were kind of some of the questions, challenges that we asked ourselves along the history? I had mentioned, interest in stem cell science and tissue engineering, led me to work closely with our scientific founders and understanding, what might be the application space, really working with pharma, what can't we do today and where can technology, provide solutions. That led to company formation and seed financing. And for the first couple of years, we were really focused on just how do you recapitulate human biology in a dish? And how do we differentiate this from other technologies? How do we set Tara apart? And really our ethos was, how do we create the most complex, most human relevant biology? And from there, with additional financing, to really build out the team, looking at expanding the platform, looking beyond healthy tissue, going into disease models, and moving into different endpoint measurements so that we can really have a platform that can tackle sort of any question or problem that people are facing. And for the past couple of years, where we've really been focused is being able to create disease models of the heart, which has led to a growth in drug discovery applications, and more recently, Tara focused on developing some of its own programs. So that gives you a little bit of flavor of the trajectory of the company. So I'll just focus on a couple of the key components throughout the rest of the talk. We'll talk about the platform, kind of how we use that platform for drug discovery and development, and then how we're using that platform for our own programs. So key attributes of our platform, it's human, it's in vitro, it's phenotypic, meaning we're looking at function, it's integrated, meaning you can measure many things at the same time, it's personalized via the ability to use stem cell derived cardiomyocytes, which helps with the translatability. And we have spent a lot of effort figuring out how to scale this. So it's one thing to make a couple of heart tissues. It's another thing to make hundreds or thousands of them, which has also established a unique proprietary IP portfolio. So what do? We can take iPSC-derived cardiomyocytes, which I think probably some of you know what that means. And for those of you who don't, it means we can take a simple skin sample or blood sample. One can then reprogram those to stem cells and then differentiate those into different cell types. And so we take the heart cells that have been differentiated from the stem cells, and we use that as our starting point to develop heart tissues. So the heart cells on their own, while they're a very, very important tool, they lack kind of human physiology characteristics. So what we do is we take the cardiomyocytes, we put them into our platform, and through a biomimetic electromechanical stimulation protocol, we form them into the tissues, and I'll play the video again. They self-assemble around these polymer wires. They're three-dimensional, they're fully suspended, and you're looking down, they're about 3 millimeters long. And then we paste them through electrical stimulation, and over time mature the individual cells into functioning muscle tissue. We can then video the functional characteristics of these tissues, and then measure the different parameters as it relates to contraction and relaxation. We can also measure calcium handling and electrophysiology and other attributes. I won't get into too much of the data here. I know this is busy, but what to really look for is the data in blue is the functional information before the cells go through our maturation process, and in purple is the result after our maturation process. So what you can see is that we generate a very nice positive force frequency of the muscle tissue. We reduce the spontaneous beating and increase the post-rest potentiation. We have a much more human relevant action potential. And in terms of structure, you can see a nice, alignment of the cells after maturation compared to before. Happy to go into this data in more detail, but the point here is that you really need to get the individual cells functioning together as a mature tissue to really have confidence in what a drug response might look like. So this is some work we did with GSK, looking at a panel of known drugs with known human response in key pathways in the excitation and contraction mechanisms. And what you can see here is that not only do we get a response and the right response, but we also get the relevant EC50 or IC50s, meaning that our tissues can really pick up the relevant human dose, which is unique among other cardiac platforms. We can also make disease models of heart tissue. So this is an example of a mutation in myosin, the R43Q mutation. And what you can see is that we can generate the hypercontractile phenotype, and then we can also get the pharmacological response, both with isopaterenol and mavacamten. And for those of you who are familiar with myocardia, mavacamten is the drug that they have moved through the clinic. And so we get the same responses that they're seeing in the clinic. So again, the ability to model disease and predict pharmacological response is what's key here. So just in terms of like platform expansion, our goal and what we've really been able to tackle over the past several years is to develop many types of tissue models. So whether they're healthy cardiac models or models generated from gene-edited cells or patient-derived, we can also environmentally induce disease models, and we can also look at chronic drug exposure. And then at the same time, increasing our ability to make many different types of measurements so you can get a complete picture. So I mentioned contractility, electrophysiology, and calcium handling. We can also look at structure, molecular profiling, and really kind of any biomarker of interest. And again, this is like a mix and match. So depending on what disease area you're looking into and mechanism, really having to be able to have a full tool set to investigate that. The platform has been published, originally in Nature and Cell from our scientific founders, and more recently, papers from Tara and our collaborators. And so that's been important as a growth during the growth of the company. So we can make these tissues, we can measure different attributes. What do we do with this? We can really apply the platform along the entire continuum of drug discovery and development. Everything from looking at target validation, because we can modulate gene expression in the tissues and look for functional response. We talked about disease modeling and how you can use that for compound optimization. At the same time, you can continually look at risk assessment and cardiac safety. And because of the iPSC technology, you can really start to look at patient segmentation and understanding which patients might really benefit from a certain medication and which really may not or might be unsafe. As I mentioned, we've worked with many different pharma companies, and what's been really valuable about this is it really gives us a feeling for kind of the breadth of applications that that the platform can tackle, and it also gives us insight into where the big technology gaps are, and it helps us to kind of like focus our R&D work in areas of high unmet need. And this is just some data on project growth over the past several years. And what you see is that as we've been developing more disease models, we can really see the increase in uptake in projects around drug discovery. And so along those lines, a couple of years ago, we started thinking about how might we tackle our own therapeutics pipeline. We've decided to very much focus on heart failure, and in particularly cardiomyopathies. We believe where the Tara technology really plays here is a great analogy, from genotype to phenotype, as successfully played out in oncology, where if you can really mimic the phenotype, you can develop a screen. And so for cardiac disease, that's been missing. But the more we understand about the genetics, the more we can mimic those diseases. And the Tara technology really allows us to develop the phenotype of, in particular, HCM and DCM models. And we can also look at genetically based arrhythmias. And so we've sort of taken a landscape approach to genetically defined disease, to focus on certain disease models for our own compound discovery and pipeline building. And so that's really kind of like a quick arc of Tara. But ultimately, we're really, believe that our platform can change the paradigm of the current drug discovery, long time, high cost, low probability of success, and very much streamline that, but again, really increase the probability of success because we can generate the human physiology in the lab. And so thank you very much, and I'm happy to take questions.